Hello. Hello, everyone. So I just found out today that I'm the president of the jury. So I'll do my best to introduce the proceedings. So first of all, for the purposes of this defense, you are in the University of Strasbourg, just for the duration of the defense. And so today, we're going to have a presentation by the candidate, Téo BARRACHO, and well, you can see the thesis title up there, Demographic and Behavioral Responses of a Polar Species to Environmental Variability. And first of all, I'll just introduce myself and the other members of the jury. So I'm Richard Phillips. I'm from British Antarctic Survey, and I'm the head of the predators group there. And I work on albatrosses and petrels.
Hello, also from me, I am Sandra Bauhaus. I'm the scientific director of the Institute of Avian Research in the north of Germany, and I run a long-term study on common terns.
Hello, everyone. I am Adélie Lescroel. I'm a research associate at Point Blue Conservation Science in California, and I've been working on Adélie penguins for the past 20 years.
And I'm Céline Boeck. I'm the PhD supervisor of Téo, one of the PhD supervisors of Téo. I'm the leader of the polar biology department at the Scientific Center of Monaco, and I'm a researcher at the CNRS University of Montpellier. And I'm an ecologist, seabird ecologist, working on penguins mainly in subantarctic and Antarctic area. Maybe Jean-Yves Georges can present himself.
Bonjour, I'm Jean-Yves Georges. I'm a senior researcher at Institut Pluridisciplinaire Hubert Curien. I'm working on freshwater ecosystems, but I'm happy jumping in some salty water with Téo Defense. And thanks for inviting me as an examiner.
Hi, everyone. My name is Nicolas Leconte. I'm professor in ecology at the Department of Biology at the University of Moncton in New Brunswick, Canada. And I'm quite glad to have this defense going on today. And we have been working very hard to make this a very nice presentation and work. We are very also focused on understanding what are the changes in the ecosystem happening in polar areas in our lab, and the focus mostly on birds and their predators. And I wish I was there with you, but unfortunately I couldn't join you. So I hope we will have time through the questions and also after to interact as much as possible.
Okay, thank you. Well, and thank you very much for the other examiners introducing themselves. So the format is there'll be 45 minutes of talk from Téo, and then they'll move on to 30 minutes of questions from myself and Sandra as rapporteurs, and then 20 minutes of questions from the other jury members, and then 5 minutes from the supervisors. So over to Téo. Thank you.
Yeah, thanks to you. Well, thank you everyone for being here today. It really means a lot to me. And so, yeah, during this presentation, we're gonna talk about how Adélie penguins in Antarctica respond to variability in their environment and notably sea ice. But before we dive in a little bit more with the penguins, I couldn't resist to quote this sentence, which says, "Who could have predicted the climate crisis?" Well, it's a bit ironical. I have to see it this way. But yeah, do you know who said that? Yeah, well, hopefully not someone with responsibilities. But jokes aside, the reason I wanted to quote this is because it makes me think of a question we may not think about enough as a society.
Téo?
Yeah?
Excuse me, but we don't see your slides moving. We only have your first slide on our screens.
Okay. I think the régie is trying to fix this.
Thank you, the régie.
Or is it?
Prima
Okay.
Thank you.
Yeah, it's
Should work.
working.
So if I do the joke again, it's not going to be funny, but okay. So yeah, just for Jean-Yves and Nico, I was saying, hopefully not someone with responsibilities. Anyway, so do you see the slides going, Nico and Jean-Yves?
Yeah, it's all good for us.
All right, great. So yeah, I was saying the question that this quote brings us to is why? Why do we need to predict? What does it bring to society to be able to predict what is going to happen? So having one supervisor from Brittany, where there are a lot of fishermen, and another supervisor from northeastern Canada, I thought I could give you the example, tell you about the example of the cod stocks in Newfoundland and Labrador. And what happens there, if you look at this time series of abundance, it's an index, but it's the idea, so the amount of cod in the area, you can see that in the 90s, there was a real population collapse. And the consequences of this collapse of the stock on the economical and social environment there were catastrophic because Newfoundland lost around 10% of its human population due to emigration after that crisis. But the main thing that is really interesting about this example is what happened actually in the decades before this collapse. And what was observable already as early as the 60s is that the size of the cod that were caught were smaller and smaller. And this conveyed the idea that there may be some signals, some hints that something is going to happen that we can observe before there is a collapse or before there is a strong change in population size. So what do we want? What is it that we want to predict when we research biodiversity in ecology, in our population ecology more specifically? Maybe it's usually the key metric is population size. So to tell you about this, I'm going to show you an example of black-browed albatrosses in South Georgia. So it's just a little population within a bigger one. Yeah, it's not chosen randomly. So this is a time series of population size. So how do you make that? You go at the place a few times, and every time you go, you just count the individuals or the pairs that are there. And what we are also very, very interested in with this metric of population size is to see how it changes through time. So for example, here it decreases, and this kind of information on the change in population size is what we use. We can use also other things, but that's one of the key metrics we can use to classify a species as threatened or not. So now we can observe that there is a change, but the thing we want to know is why. How is that changing? An approach to answer that question is to decompose. So we have here the first step is the population size. But the first decomposition of that we can make is that we can decompose it in values, in quantities that are called vital rates. So among these rates, you have the survival, either of the adults or the juveniles. You can have immigration and emigration, breeding propensity, so how many individuals attempt to breed. And you can also have reproductive success, for example. And if we keep decomposing that, behind every vital rate, there are actually individual traits that can be behavioral or physiological that are going to affect these vital rates. So for example, the physiological traits can be size or mass. For example, in fishes, the biggest fishes also make the most eggs, or it can also be behavioral. So for example, in seabirds and in birds in general, the duration of the foraging trips, for example. And if we keep going up the cascade, then we find the environment that is often responsible for this variability. And so here, for example, we have the increase in sea surface temperatures since the 1900s. So now you can start to see the idea of a cascade between the environment and population size. And to exemplify this a little bit more, I thought I could tell you about a real-life example, and actually one on yellow-bellied marmots in the US. So in this population, what the people that were working on them saw is that there was a huge increase in population size after the 2000. And they wondered, why is that? Why did the population increase? And at this point, it's anecdotal, but they used a model called an integral projection model to try and understand the cascade that was responsible for this increase. So the first thing they realized that this increase was driven by higher adult survival, that was itself driven by higher body mass before the winter season, that was itself driven by springs occurring earlier and earlier due to climate change. So now another question we may ask, because this is really interesting, this detailed knowledge, but it's super useful for understanding. But the question is, can we use it to improve our predictions, to improve our forecast? And when we say better forecast, better prediction, what does it mean? It means forecasts that are both timely, so at time scales that are interesting to us. So it can depend, it can be short or long, depending on the questions, and also reliable, so with as less uncertainty as possible. So the framework in which I wanted to tell you to present the things today is the one that is aiming at doing just that, using this detailed knowledge to try and predict. And it's a framework called the timeline to collapse. But we'll see that it can also be used for other things than just to predict collapse. So it comes, I think it would be interesting to do a bit of history. It comes from the early warning signal theory. So if you consider that this is a time series of abundance, population size, this theory says that you will have statistical signals, statistical artifacts before a decline that can be used as early warnings. But the timeline to collapse framework is a development of this theory that use multidimensional signals. So what do we mean by that? So it's really also something I should precise is that it's particularly focused on ramping stressors. So for things that stress to the species or individuals that are increasing through time. So for example, here, a decrease in food abundance. So the framework says that the first thing that is going to change in response to the decline in food abundance is going to be either the behavioral or the physiological trait. So for example, here, an increase in trip duration. Then we will see a signal in the vital rate, so for example, the reproductive success, and then only ultimately at the end, we will see an effect on the population size. And this framework really emphasizes the idea of looking at this sequence of consequences on different values, different metrics. And so, yeah, we were saying why this framework? Well, the first reason is that it can be useful. 50% of the reason is that it can be useful for predicting decreases mostly, but also increases. But also, and that is really important, I'd like to point your attention to that, is that it's very useful visually to understand the links between environment and changes in population size. So what do we have of validation? It's quite a recent, I mean, the ideas behind the framework are not recent, but the framework itself is quite recent. And we have partial laboratory validation of this framework on paramecium in microcosm. So here they exposed the paramecium to a stress at H equals 0 hours, and they observed the decline in population size at 111 hours. And what they were able to show is that the change in behavior, so it's the swim speed of the paramecium, that started to be informative about the decline in population size at 73 hours. So compared to the lifespan of those organisms, it's quite an early signal. And they didn't test the vital rates. That's why it's a bit still in validation. But the question we want to ask today is, can we also apply this interesting framework to wild populations? So for example, in the case of today, Adélie penguins in Antarctica. So you may think, if you know a bit about Adélie penguins and other seabirds, you would know that they are long-lived. So that means they, for example, in the case of the Adélie penguin, they can live up to more than 20 years. So that's not really practical for studying these things, especially with the fact that they breed in Antarctica, which is not really accessible. But the reason why it's very important and really interesting, it's also about the context for the Adélie penguin. So here you have all the colonies around Antarctica, the breeding colonies, and what we see is that the population trends are contrasting across this region. So with decreases, for example, in the Western Antarctic Peninsula, or increases, for example, in the Ross Sea. And our current predictive abilities for these changes in population size of Adélie penguins are at large scales, so the scale of a region, thousands of kilometers, and long time scales, so several decades. We can kind of do it with a lot of uncertainty, but at shorter term, on shorter time scales, and for local population, it's really difficult. So there is really a use for a framework that can do that. And also an important point for this species is that it's really linked to sea ice, which is a defining element of the Southern Ocean ecosystem. All right, so now on to the plan of the presentation today. So the more focused question that we're going to try and answer is, how is environmental variation affecting the population trends and the population dynamic of Adélie penguin? And to do that, we're going to look at 3 different vital rates for which we will try to understand the links between all the components, between the environment and the population size. So these rates will be reproductive success, juvenile survival, and adult survival. And for each of those rates, we will try to build this kind of timeline, this kind of sequence, to understand, to pinpoint gap, and maybe to predict what will happen to population size. And also something that is going to come back throughout the presentation is the idea that we will be looking for general patterns. So by comparing data with other population around Antarctica to aim for generality, for understanding the mechanisms that may be true at the species scale. Before we go on to the results, there are a few information that you have to know about the Adélie penguin and its environment. And the main one is the seasonal cycle of Antarctica. So here you can see the seasonal cycle of sea ice with huge increases in winter and very rapid decreases at the onset of summer. That's what you can see here. And the breeding cycle, so the reproductive activities of the Adélie penguin, are based on that seasonal cycle of sea ice. So we won't go into too much detail, but basically the Adélie penguins, they do their reproduction only during summer, so when sea ice extent is the lowest. And a phase of the breeding cycle that is going to be of interest to us later on is the chick guard stage. And during this stage, you have always a parent present at the nest. It's right after hatching that guards the chick, and another parent that is at sea foraging for food to feed the chicks, the chick or the chicks. And yeah, another key idea, key metric that we'll have to keep in mind for the rest, and after that, that's it. It's called sea ice concentration. What is sea ice concentration? We may say SIC later on. It's a value that we can give to an area. So for example, this pixel here, and that defines how much, what's the percentage, the proportion of this area that is covered by sea ice. So it's a measure we use to quantify how much sea ice there is in an area. All right, so I just told you that Adélie penguins were associated with sea ice, but how does that manifest? So what we have proof that, or evidence that at large scale, as I was saying earlier, the population growth rate, so how much are the population increasing, decreasing, or being stable, is correlated with the winter sea ice. But what's underneath this correlation with the population growth rates are actually correlations that have been made between the penguin vital rates, so survival, reproductive success, things like that, and sea ice measures. And one thing that is really interesting, and a word that I really like, and an idea I really like, is that this association between the penguin vital rates and sea ice can be quite specific. So it can have particular spatiotemporal windows, and types of sea ice may have different impact. So that's what you can see here. If you consider the sea ice at different scale, it may not give the same results. And I think this idea is embodied by a term coined by Emerson and Southwell in 2022, which is penguin relevant sea ice. Well, it's a bit catchy, but it's just to convey the idea that different types of ice may have different effect on different vital rates, for example. All right, so now how do we measure that? How do we measure vital rates in penguins in the wild? So this is the setup we used for the thesis, this long-term monitoring setup in Adélie land. So it's located south of Australia. And so it's an archipelago, the Point Geology archipelago. And on this archipelago, there is a breeding patch, a subcolony located here that is monitored with PID tags, so RFID tags, which are really small. And this is the same kind of tag you could put in a dog or a cat elsewhere. And so the subcolony we monitor comprises around 270 pairs. So it's quite a lot of individuals. And at the bottom of the colony here, you only see one. There is also one at the top. We have RFID way bridges. So every time the penguin that is PID tag crosses the bridge, it's in identification, but also we have data on its mass, on its body mass. All right. So now on to the first chapter, reproductive success. And the first question, the general question for this chapter is how foraging behavior links sea ice to reproductive success. So to try and decompose this relationship. So first, a bit of context. What do we know on Adélie penguins that is important for this? So Adélie penguins are central place foragers. So this is an important term in ecology, and it describes individuals or species that have to come back to the same place when they go foraging. So here, the Adélie penguins, they are constrained, they cannot use all the sea they want because they have the constraint of returning to their nest to feed their chicks. And the reproductive success of this species, we usually calculate it, we estimate it as the number of chicks per breeding pair. So we count the breeding pairs at the beginning of the season, and then we count the chicks at the end. And it usually varies between 0 and 1.5. And so this reproductive success, because of the central place foraging constraints, it is influenced by sea ice, by the amount of sea ice in the foraging area. And so by food availability, that is linked to sea ice. And food availability, it can be decomposed in food abundance, but for this, we don't know a lot. But what we know about better is about accessibility. So in situation, So in situation like this, you have here the colony, you can pay attention to the scale. When there is no fast ice, so this type of solid ice, the penguins, they can access the open water freely. They don't have to walk over solid ice to go foraging. And in other situations, there is fast ice like that. They have to commute over this fast ice. And this we know it plays a big role in driving their reproductive success. And basically the idea is that when the sea ice is like that, when there is fast ice, their reproductive success is very low. They don't manage to breed when it's like that. But what do the climate prediction tell us? Well, there is a lot of uncertainty in sea ice forecast and sea ice prediction, but from the one we have, so this is from a very recent preprint from this year, and I'd like you to pay attention only to this curve here in light blue. What this forecast is that in summer, the sea ice around Adélie land, this is for Adélie land, so quite relevant, is going to decline. So yeah, we know, and it's the sea ice, but also we have other evidence that it's mostly the fast ice that is going to decline. It's it's also a recent paper here. So the question becomes, now we know the impact of fast ice, but what we would like to know now is what is going to happen when there is no fast ice, if there is really less, not a lot of ice, or if there is a little bit more ice, but still no fast ice. So that's the first objective of this section, to know what happens when there is no fast ice. And the second objective is to try to do this decomposition that I was telling you about between the environment and the vital rate by using also the behavioral or physiological trait. So right, onto the methods. So for this, we used the long-term study at its full scale with the data between 2010 and 2024. And the metric, the foraging metric, behavioral trait we focused on is trip duration. Why did we choose this one? So trip duration is just the time it takes for the individual to exit the colony, to come back and to feed the chicks. So this is really accelerated. This is a 10 days period that you can see here. So no worries, they don't go this fast. And yeah, the reason also we use that trait is because it can be obtained with minimal invasiveness. So for example, for our birds, the one that we monitor, we implant them once in their life with a pit tag, and then we get a lot of information, such as this information. So for this in the team, it's really our basic information. And you can see very well in orange, you have the periods when the bird is on land, and in blue, the periods when it's at sea. And you can see very clearly the different breeding phases. So you have the incubation, the chick guard, and then the crèche. But today, for this talk, we're going to focus only on the guard stage, and the reason for that is because it's during this stage that nest failure peaks, so that the most nests, they fail. So it's probably a very important phase. And the amount of foraging trips of data that we gathered for this phase is in excess of 23,000, so quite a lot of foraging trips. All right, now what happens? So the first question, if you remember, was what happens when there is no fast ice to reproductive success? So a direct relationship between the environment and the vital rate. So what we see here is that we find again this negative relationship that we knew about a bit already. But what we can also see, if you look only at the fast ice absence scenarios, is that the relationship is maintained. So that's quite an important result. And the side result of that is that when there is almost no sea ice in the foraging area here, the breeding success is actually very high. So the Adélie penguins, a bird that is associated with sea ice a lot, they are actually able to tolerate a near absence of sea ice in their foraging area during summer. There is really this aspect of periods that we have to keep in mind. All right, now if we try to decompose this, to break it down a little bit. So first, the relationship between the environment and the behavioral trait. We can see that when the sea ice concentration in the area increases, the duration of the foraging trips that you have here and here, the sea ice, it also increases. So a positive relationship that is also maintained when there is no fast ice. And now the last part of the decomposition, the relationship between the behavioral trait and the vital rate. Well, we also see this significant and in this case negative relationship where the reproductive success decreases as a guard stage trip duration increases. So here, what this means is that we have a fairly simple to obtain behavioral predictor of reproductive success, and that is quite interesting to us. But to make it even more interesting, what we were interested in doing was looking at it at the circumpolar scale around Antarctica. So lucky for us, that's what we were able to do, because there are a lot of polar programs, a lot of nations that also collect this type of data that we could put together and try to see what happened to this relationship between trip duration, so it's guard stage, but also a bit from the crèche stage. It's not really perfect in that regard. But what we can see is that the relationship is broadly maintained, the relationship between productive success and trip duration. So it's an indicator. So if we look a bit at the overview, having this relationship validated at the circumpolar scale is really important because it increases our confidence in using this behavior demography linkage in the future. Because likely, I mean, in the future, the condition may be different, but the conditions around Antarctica are already quite different. And so, yeah, for the overview, we showed also that all this relationship, all these links in between were maintained when there is no fast ice. And if we try and build the timeline to collapse, as we said we would do at the beginning, it's where the usefulness of the visualization aspect of the timeline takes all its importance. As you can really see very well here that when the sea ice increases, the trip duration increases and the breeding success decreases. So that illustrates the usefulness of the timeline to visualize. In this case, just to visualize and understand. All right, so what happens after the breeding season? Well, after the breeding season, when young penguins are produced, they go at sea. And after that, the vital rate that matters is their survival. So we call that the juvenile survival. And the question we ask here is what is the temporal and also spatial variability in juvenile survival of Adélie penguin, and what may be the drivers of this variability? So first, for juvenile survival, it's really an interesting vital rate because it's quite mysterious, actually. So for when Adélie penguins leave the colony for the first time, they may not come back for another 2 years. And we have very limited information on what they do during those 2 years. So actually, we only have 2 studies that put loggers or trackers on those birds, and they showed that they could move thousands of kilometers during this period. And this lack of knowledge on what happens to juvenile is quite contrasting with the importance that this vital rate may have on population dynamics, which is this part, if you remember, of the timeline. And we have evidence for that, solid evidence for that for other bird population, including seabirds. All right, so our objectives for Adélie, so here for point geology, were to quantify the temporal variability of juvenile survival, to try and find and discover and uncover its environmental drivers, and also to do this cross-population comparison with information we have from other colonies. So how do we measure survival using the setup that we presented earlier? So we use what is called capture-recapture data. Well, in this case, it's electronic, but it doesn't really matter. And how does capture-recapture data looks like? So it's when you go every year at the colony and you assess whether you detect an individual or not. When you don't detect it, you put a 0. When you detect it, you put a 1. So for example, this one is probably still alive, and this one is probably dead. Well, this information, we do it for a lot of individuals, so in this case, more than 3,000. And then we put it in in capture-recapture models, in this case, multi-stage models. We won't go into the details here. And the output, what's really interesting is the output of those models is annual survival rates, in this case, specific to the juveniles. And so we were able to obtain those estimates between 2007 and 2020. And what we can also do with this kind of models is to try and assess what are the statistical relationship between juvenile survival and a set of what we call environmental covariates. So in this case, sea ice and other environmental parameters that we suspected would have an effect on juvenile survival. And another aspect I really wanted to tell you about all this is the strength actually of our study design. So usually, in many systems, you have here the main study site. You would try and detect only the birds that come back to this study site. But what happens if there is a juvenile that fledges from here, but only come back and decides to breed in this colony, for example? Well, you may miss it and you may estimate survival not really accurately. So what we did, what was started in 2013, was that mobile RFID antennas were deployed across the archipelago. And this was really useful to control for a short distance dispersal. All right, so now on to the results. So we're going to see a time series of juvenile survival, and that is this one. And the first thing that we see looking at this time series is that there is a lot of interannual variation. So it varies a lot, but despite this variation, what we also observed is a 2.5 annual decline in juvenile survival, which equates between 2007 and 2020. This equates to a 32% decline in the survival of young penguins. And this is on top of that, on top of this decline of juvenile survival, you see that there is no data point here. It's not for no reason. It's just because on those years, there were extreme sea ice events during the breeding season, and no juveniles at all were produced in the entire archipelago. Maybe one in 1 year, but it's as if it was nothing. All right, so now we wondered what is responsible for this variability. So that's where the environmental covariates come into play. I'm sparing you the ones all the ones that we studied, but I'm just showing you the most significant, the most important ones. So what we found is a positive relationship between the sea ice concentration in April. So April is the moment right after the fledging, they leave the colony, and the juvenile survival. So that means when there is more sea ice in the area, in the vicinity of their natal colony, when they fledge, they survive better. I mean, that's the correlation we find. And this is also in good accordance with previous results showing that this post-fledging period is very critical to juvenile Adélie penguins. But we are not satisfied with just this general relationship. And if you remember the term penguin relevant sea ice, we wanted to understand it a bit more. And what happens is sometimes, again, pay attention to the scale, sometimes the environment in April around the colony looks like that, so quite dense sea ice with those areas of open water that are called polynyas. And in other years, it looks like this, so much less dense sea ice with a lot of open water in between the ice blocks. And what we found by looking at this in more details is that there was a positive relationship between the survival of juvenile and the amount of sea ice in the area that was heavy sea ice, so compacted, dense sea ice. And we found also the opposite relationship, that there was a negative relationship with the amount of loose sea ice, of fragmented sea ice. So that goes on to illustrate well, I think, the term penguin relevant sea ice. All right, now we've identified potential environmental drivers. A point we were also interested in: what are the demographic, what are the population dynamic consequences of this decline? Well, to that, we can gain insight from looking at what happened at the other colonies I told you about a little bit already. So the first study to report time series of juvenile survival was one made by Americans, Hinck et al. in the Antarctic Peninsula, so here. And what they found is that juvenile survival at the study site, it declined by 1.3% a year between 1982 and the year 2000. And the consequences for this, here you have population size, and the same scale here. The population, it declined after that. And maybe if it was one population, that wouldn't be really significant. But what the Australians, Emerson and Southwell, observed a few years later, a time series that is a bit different here, they also observed a decline, and that was also visible in the population size. So what's happening at Point Geology with our decline in juvenile survival? Well, quite surprisingly, what we see for now is indeed a decline that is of a magnitude that is comparable, if not higher, than at the other places. But this is the population size. So the population size at Point Geology is not decreasing at all. And I'd just like to point out that the time series here stops in 2019. So, I mean, other people have the data, and that would be interesting to see what's happening now. All right. So how can we explain that there is no decline yet at Point Geology? Well, the first aspect, again, we will look at the other study sites. The first aspect that is really important to consider carefully is that the time series at other sites, and yeah, again, this is not the same time period exactly. Yeah? The time series at the other sites are longer. So maybe there needs to be a longer time period during which juvenile survival declines for it to generate an abundance decline. And also what we may understand, we may see from the Bécharvez Island, so the green one, the green population, what we may see happening is that there is actually kind of a threshold or a delay, between the moment juvenile survival declined. So here, 10 years of decline in juvenile survival, but the population kept increasing for 10 years. So that illustrates something that correlates well with the idea of the timeline to collapse, is that there may be delays in the moment a vital rate starts decreasing and the moment the population size starts also decreasing. So if we construct it here for juvenile survival, you see that the behavioral or the physiological link between the environment and the vital rate is lacking, and we will discuss about that more at the end. All right, now on to the last vital rate that we will examine today, adult survival. And the question here, the focus for this part will be more on the consequences of extreme events on this vital rate. And adult survival, yeah, I'm always a bit interested in this vital rate because the theoretical model, so in theory, what we know of adult survival in long-lived species is that it should theoretically have big impact on population dynamics. And a consequence of that is that it often shows low variability. So if you imagine here a time series of adult survival, you have adult survival in time here, usually it looks like that. So really stable, really flat. It doesn't vary much because it could affect the population dynamics too much. But what we know recently more and more from extreme events is that, yeah, sorry, something I forgot to mention is that when it's flat like that, we say that it's buffered. The theory that says that it should be flat and show low variability is called demographic buffering. So that's why we say it's buffered. But extreme events, so events that happen only that rarely happen, individuals may not see a lot of time in their life, they can overcome this buffering. So how does a time series of adult survival look when demographic buffering is overcame? Well, it looks like this. So it's mostly flat and then it drops. And the reason I'm telling you about this is because this kind of black swan events, that's how they called, we have a recent study from 2022 that says that they may have a key role in driving the population dynamics of Adélie penguin. And what's also important about this study is that this is a simulation study. So what we would be interested in is validating this simulation with real data. And so the reason we are able to ask this question about the impact of extreme events is because at Point Geologie, at our study site, we actually had 2 extreme events only in the 2010. So what were these extreme events? So these were extreme for the specific period, so for January, and usually it looks more or less like this, but in 2 years, 2014 and 2017, it looked like this. So a lot of extensive summer sea ice. So it's a specific type of extreme event that we are looking at. And what this extreme event caused were massive breeding failures. So in 1 year, the whole archipelago, so it's a population of around 40,000 breeding pairs, they produced no fledglings at all. And in the other one, they produced only 1. So 1 fledgling for 40,000 pairs, it's not a lot. And that's what we can see here. So 2 black swan events for reproductive success. But the question we asked is do these extreme events, do they affect also, on top of affecting reproductive success, do they also affect adult survival? And because adult survival is supposed to be buffered, how can it be that such events, they affect this rate, whereas it's supposed to be buffered? So the methods are really the same that we use for juveniles. It's capture-recapture analysis, a very similar model. And what we wanted also to do for this vital rate is to control for potential covariates that could explain variation in adult survival, because we don't want to have confounding effect of those covariates. And so we didn't select them blindly. We had other studies pointing towards some specific covariates. And we also used information on the movements of the adult birds in the winter season from other studies to select, if you remember the figure from the beginning, to select the most relevant spatiotemporal scale to measure the environmental covariates. So, all right, what did we find? So these are the 2 extreme events, and this is the adult survival time series. And what is striking right away when you look at this is that in 1 year, in 1 extreme event, so the one from 2014, adult survival didn't drop. It wasn't significantly different from the other years. But in the other extreme event year, so 2017, we can see a 15% drop in adult survival. So it may not seem like much, but for the adult survival of such a long-lived species, it's actually quite relevant. So only 1 black swan event for adult survival. All right, so what about other drivers I told you about that we wanted to control for? I'm only here showing you one, so the concentration, the amount of sea ice in the molting area, so where birds change their feathers. And you can see here, it's well illustrated that it cannot be those other drivers that generated the pattern in 2017. It sits really outside the distribution of the other survival estimates. All right, so how the second question we wanted to ask is how can this be? What can we can we infer from what we observed to get a bit more idea about the mechanisms? So this is, yeah, the two contrasting years. And yeah, I told you that those years were both extreme, but actually when we look at them in more details, we realize that there are some differences between them. And notably, one of those important differences is that the access to open water was much shorter in 2014 than in 2017. So our hypothesis, our speculation about this is that in this year, when the birds, they failed the breeding, they stopped breeding and they wanted to go back to sea to feed, to replenish their energy reserves, they had to cross a distance that was too much for them, too much for the energy reserves they had left compared to 2014. So this represents what we believe is an immediate cost of attempting to breed in a year where it was this difficult to breed. All right, so now for the overview of the adult survival part. So as we just told, we gained insight on the how extreme event may disrupt the adaptation of long-lived birds to breeding. And also what's important, and is not shown here, is that we showed that extreme summer sea ice events, extreme ice events, in summer, they can affect the adult survival, but we have evidence from other colonies that there are drops in adult survival that are not linked to extreme summer sea ice events. So there may be a role for winter events. And as for the other sections, if we build the timeline here, well, we see that we also have a missing link, but this is ongoing. We'll talk about it right after. And we also see the limits of this framework, this timeline to collapse framework for predicting when the events, when the disturbances, the stressors, they are pulsed. They are not ramping stressors. And so for this, the framework is not really adapted when the vital rate is driven by events such as black swan events. All right, so now onto the synthesis. How do we bring all this together? How do we synthesize all this information and these detailed links? So we can first start with a retrospective analysis, and this means answering the question: is the point geology population, facing impeding changes or impeding declines? Well, to try and answer this, we can build the timeline for reproductive success, for juvenile survival, and for adult survival, and that ultimately affect the size of the population that we have from collaborators. And well, actually, for the visualization and understanding aspect, well, it's undeniable that we have gained more knowledge by identifying these links, more detailed for reproductive success and maybe less detailed for the survival probabilities. So that's for sure, because we're able to visualize. And we also, a really nice use of this framework is to be able to visualize the lags, as we discussed for juvenile survival by looking at the other populations. And what's also interesting of the idea of using this framework with several timelines, several vital rates, is that we may be able to see offsets. So for example, this has been shown in Titre, I don't remember which, great Titre, I think, in Europe, when you had one vital rate that was decreasing, but another one that was increasing at the same time. So overall, the population remained stable. So, but here, the main problem we face, and that was, I mean, we kind of knew about it at the beginning because the species is so long-lived, is that the time series length, the duration of our study may not be sufficient, even though collecting all this data for already almost 15 years takes huge amounts of money and of work. And also something that we may be closer to solve is that we would need this information on the behavioral or physiological components of the timeline to have better predicting abilities. All right, so that was for the prospective synthesis, but we can, the retrospective, sorry, but we can also do a prospective one. And this means the prospective synthesis, answering the question, we framed it this way: how many extreme events, such as the one we discussed, is the population able to withstand, able to manage? So yeah, this is an ongoing thing, an ongoing project that uses matrix projection model. So again, I won't go into the math details, but these are just simulation tools that use the data. So this is how they are constructed, and this is the data they use, and this is the data we obtained in the previous chapter. So if we put all of this in a model, we can simulate different extreme event scenarios. And in this case, we simulated two such scenarios. The first one, where the extreme events, that's the previous knowledge we had before this thesis, affects only reproduction. And the second scenario, where extreme events, that's the new knowledge we have, they can affect reproduction, but also survival. All right, so what do we find? So here you have the population growth rates, and basically when the line here goes below 1, it means that the population decline. And here we have the extreme event frequency, so 1 per decade, 2 per decade, and so on. And what we see is that in the first scenario, where extreme events affect only reproduction, the curve goes below 0 only after around 3 events per decade, whereas if extreme events affect both reproduction and survival, then the population starts declining as soon as the frequency of extreme event reaches 1.5 events per decade. All right, but this was super, I mean, to me, it's super interesting, but this was still very local, very focused on the point geology population. So what happens if we try and synthesize and scale up the synthesis at the scale of Antarctica, and we gather all the demographic, I mean, most of the demographic information that we can have around Antarctica, and we can present it like this. So this is maybe a bit not so natural to look at. It's the density distribution. So you just estimate, for example, adult survival, and you see how this distributes on a scale. So here you have 0% adult survival, here 100%. So, for example, the way to read this is that Admiralty base of this colony, the adult survival probabilities are usually lower than the ones at other sites. And we can do that also for the other vital rates, so reproductive success and juvenile survival. And what does it brings us to be able to compare those colonies, to compare those populations, is that we can pinpoint which vital rates are most influent. And this is a very important idea. And for example, here, the fact that we know that in the Western Antarctic Peninsula, so this region, the populations are really declining, but it suggests that adult survival may play a big role because it's really different from at the other study sites. And also to have information on different populations, if the same thing happens at different regions of the same continent, the scales are really big here. Between these colonies, it's more than 3,000 kilometers to give you an idea of the scale. So if the same things happen at different locations, it gives us increased confidence and reduced uncertainty for species level forecast. All right, so now on to the perspective that we can have this information on the importance of vital rate by comparing population, just comparing the raw estimates like this. But something that I'd really like to focus my efforts on in the future is to apply some tools that are called life table response experiments, and they can allow to measure the contribution of different demographic rate for a population. And then, so for example, this is an example in Norway, and you have in blue here, it means that reproduction is the vital rate most driving population trends. And when it's in yellow like this, it means that adult survival is the vital rate most responsible for variation in population size. And so doing this, it can allow to pinpoint which vital rate are the most important. And this is really important for us to be able to pinpoint which timeline, which component of the timelines we have to work the most on. And so this is linked to the flexibility of the timeline. If we don't focus on the vital rate that is most important, maybe the timeline approach is not so useful. So you can use these ideas to progress in this direction. And yeah, the second to last perspective, as we were telling, is filling the gaps that we have here in the timeline. And a really promising things that we are looking at right now is to look at variables that we can extract from our automatic Weigh-Bridge monitoring. And so, for example, here you have the change in body mass of an individual throughout the breeding season. And one of the metrics that we would really like to be able to have is how much is body mass, so when it exceeds the colony, when it's empty, how much is its body mass changes throughout the season. That is something we are actively working on and making good progress on. And finally, now we're almost done. I'd just like to mention the last perspective. Maybe it's the more romantic perspective. Maybe it's because I'm a bit romantic. It's about biparental care. So so for the timeline to collapse, we're really focused at the population scale, so at linking behavioral or physiological metric with vital rate metrics at the population scale. But you have other models, such as the integral projection models, or IBM also, the other types of model, that use relationship between an individual trait and an individual measure of fitness. But the problem with biparental care species is that you have indeed this relationship. So the performance of the chick, the mass of the chick, the number of chicks is going to be influenced by the first partner, but it's also going to be influenced by the second partner. And this is something that we are looking at the moment. And also how, so for example, using the data on body mass that we just showed earlier, and how the body mass of the male partner and the female partner may be managed in tandem between those two. All right, and yeah, to conclude, just before the acknowledgements, I'd like to well, it's a bit of a catchy sentence, but just it's a bit of semantics maybe, because I used to mistake prediction and anticipation, but the idea here is we can have all the best prediction, all the best forecast we want. If we do nothing with it, and doing means anticipating, it doesn't really matter. And this decision of anticipating, doing something with the forecast doesn't depend on us. I mean, on us researchers, it depends on the politicians. All right, and so I'd like to thank you all for attending today, and especially the thesis jury, so Sandra, Richard, Jean-Yves, and Amélie. And also I hope you can notice the pun here, my supervisors, Céline and Nico. I'd really like to thank the French Polar Institute, that is the organism responsible for all the logistics in Antarctica. We couldn't collect this amazing data without them, especially the missions of the Adélie and Terre Adélie 71, 72, and 73, with who I had the chance to work. And also all the partners and the lab I worked at during these amazing 4 years. So the Centre Scientifique de Monaco, obviously, thank you a lot, Denis and Sylvie, the Université de Moncton, actually we can say both because they speak both French and English, the IPHC in Strasbourg, the CEF Montpellier, of course, and also a lot of thanks to the Monaco Oceanographic Museum for welcoming us today. And also, oh yeah, wow, sorry, I forgot about this. All of my amazing scientific collaborators, so Thierry Raclot and Jean-Pierre Desforges, especially, that were in the thesis follow-up committee, really super useful comments from them during those 4 years. And yeah, also all the co-authors and the collaborators, thanks a lot to you. And yeah, thank you. I think that's, yeah, that was just, yeah, I mean, do whatever you want with it. And then I prepared the slide for questions.
Téo, thank you very much. That was a fantastic talk. So I think we'll move on now to 30 minutes of questions from Sandra. Thank you.